So it is absolutely my pleasure to introduce Madelyn Jamsa, AKA Maddie. For those of you who don't know, this is her last semester with ENST. And she is actually, I didn't realize, was actually a legacy Terp. She's a second generation of her family coming to Maryland. She graduated from ENSP in the wildlife program in 2022, so not that long ago. And now she's, before she came to grad school, she did some really cool, kind of jealous projects. She worked on sea turtles in Florida. She worked on humpback whales in Hawaii, all the kind of dream projects. And then she came and worked on turkeys. And so she's going to be presenting some of her master's work, which she defends in a few weeks, to us for her last seminar. So Maddie, with that,
all right.
Ready?
Thank you, boss.
Thank you.
Hello, everyone, and good afternoon. And thank you, Jennifer, for that wonderful introduction. My presentation, I believe, is a great follow-up of a seminar we had here a few weeks ago, where my wonderful colleague, Dan Herrera, who I do not see, discussed with us bird abundances in urban cities. Today, I am going to discuss with you an increasingly common but unlikely urban bird, and that is the urban wild turkey. So without further ado, I am very excited to present to you all today my master's thesis, which is Long Legs and Long Beards, examining tick turkeys' dynamics across an urban-rural gradient. So when I say wild turkey, I'm referring specifically to the eastern wild turkey, and you can see their range highlighted here in blue. In the early 1900s, in the mid-Atlantic region and in the state of Maryland, these birds were almost harvested near to extinction. However, successful reintroduction and transplantation programs have since reestablished this bird well within their historic range and beyond, earning the turkey the most notable title as the comeback kid. This reintroduction program is often regarded as one of the most successful reintroduction of a wildlife species in North America. So this reintroduction was so successful because wildlife scientists and managers really understood the brooding habitats of wild turkeys. So what exactly does their brooding habitat look like? It typically consists of this knee-high herbaceous vegetation. As you can see, this is advantageous because poults are able to feed while remaining shielded from the grasses, and the hens are able to look out and search for predators. On the other hand, wild turkeys require roosting habitats, which primarily consists of these large hardwood, mixed hardwood forests. However, turkeys are regarded as generalists with their habitat selection. They can roost in primarily anything, which consists of immature bushes, and often in urban and suburban areas, we see them roosting on people's homes. Wild turkeys, in terms of their diets, are opportunistic feeders. They feed on just about anything in the ecosystem, which consists of maybe ticks, and I will discuss this point in depth later, acorns from masting trees, bugs, grains, and as well as waste grains from agricultural productions. And particularly in these urban and suburban ecosystems, we see them feeding on bird feeders. So why should we care about wild turkey presence in our ecosystems, and what is their significance in the state of Maryland? Well, one, they are a prey species for a variety of mesocarnivores we see here, most notably the coyote. They also directly control the abundance of other species through their direct consumption. As I had mentioned earlier, turkeys feed on bugs, so by consuming bugs, they then are able to regulate the bug populations. Specifically, in the state of Maryland, they hold great economic importance. Approximately 10,000 participants every year participate in the spring annual harvest, which runs from April through May, and this brings approximately $5 million annually to the state. Socially, they are important for you and I because these are the birds that often appear in front of us at our Thanksgiving feasts, but they really resonate with these indigenous communities. Parts of the bird, specifically the feather, are used in a variety of indigenous ceremonies and rituals, and the presence of a wild turkey is often thought to bring abundance and prosperity to your future. However, with the presence of wild turkeys, there are also some severe implications, particularly in these agricultural systems. For one, turkeys perform a behavior called dusting, which is when they plop down in an agricultural area and they use their feathers to bring up dust around them. This is sort of a turkey's way of itching and cleaning themselves. However, this behavior becomes particularly destructive because the bird returns to dust in the same area every time, and these dust bowls can be approximately 1 meter in diameter. On the other hand, as I mentioned earlier, turkeys are opportunistic feeders, and they've been documented feeding on crops, which can often cause monetary losses to farmers. Particularly when we look in our urban and suburban systems, we see economic implications with bird presence. As you can see here, turkeys like to roost near roadways. So when they roost near roadways, they often increase their chance of automobile collisions. Same thing here as well. With automobiles, turkeys, like most birds, are able to see their reflection in surfaces like car doors, car mirrors, or other lawn ornaments. The turkey sees its reflection, it perceives it as another bird, and it tries to attack it using its beak or its spurs. So this can often result in economic and aesthetic implications for human properties. So I've discussed potential economic implications that we see with wild turkey presence, but more specifically, what does this look like in the Washington-Baltimore region? Well, it can look like a whole variety of things. It can look like wild turkeys being spotted in areas of high human use, like the metro station. Who here has seen an urban or suburban wild turkey, or by show of hands? Right, so quite a few of you have seen a wild turkey in an urban or a suburban in an unlikely area. You all may remember a few months ago, this news story that broke of the male turkey accosting individuals on the Anacostia bike trail. As you can see, this individual here trying to defend herself from the bird with her bike. So despite there being an anecdotal increase of sightings of wild turkeys in these urban and suburban systems, there is a large knowledge gap when it comes to their abundance and distributions in urban and suburban landscapes, which then begs us this question: why should we care about them in our urban and suburban settings? Well, there's a potential decline of rural turkeys in their more historic agricultural and rural regions. They are potentially influencing tick loads and abundances in ecosystems through their roles as tick hosts or through their direct consumption of ticks. I will be discussing this point in depth later. And turkeys are potentially experiencing a range shift. So this is thought of them because they are reaching carrying capacity in their more historic agriculture and rural home ranges, which is then causing wild turkeys to seek and shift their range to seek suitable habitat in these urban and suburban areas. And lastly, there is very little research about these urban birds. So this knowledge gap then prompted me to create these two study objectives. So the first one, which I will be discussing in depth with all of you today, is evaluating turkey occupancy and density across an urban-rural gradient. I hypothesize that what we are seeing and hearing anecdotally is true, and that turkey occupancy and density will be higher in these urban and suburban areas, particularly these suburban areas that are associated with things like manicured lawns, turf grass, as well as bird feeders, because we know that turkeys like these areas during the brooding season when they are really focused on feeding themselves and feeding their young. So in order to do this, my study area was Montgomery County, Maryland. It is located just west of where we are here in Prince George's County. It is Maryland's most populous region with approximately 1 million residents. Montgomery County is spatially unique in the sense that it really exhibits this gradient of urbanization. The southernmost region, as you can see here, that borders the Washington, D.C. metropolitan area, is highly urbanized, and these green spaces are often fragmented. However, as you move northward towards Frederick County, these areas are more agricultural and rural, and these green spaces are often more continuous. So to collect data on wild turkeys, we deployed 98 cameras throughout 8 spatially unique parks in Montgomery County, Maryland. These cameras sit approximately at hip height, which is about a meter up on a tree. They are triggered by movement in their environment to capture 3 images across a 0-second continuous interval. As you can see here, I've provided images of our cameras for folks who use cameras arrays, right? This picture may look familiar. This is a lockbox that you place on a camera. An individual comes and messes with your camera. You put a lockbox on it. You think that deters them. It does not. Right, so here's the breakdown of park, the number of cameras and the array in each park, and its urbanization classification. As you can see here, Rock Creek Stream Valley and Cabin John Regional have significantly more cameras than the remaining 6 parks. This is because these parks were set up to do a very specific analysis, which is the rest and rem analysis. But as you can see, Sligo Creek through Woodstock Equestrian here, parks typically contained an array of anywhere from 6 to 9 cameras. And so spatially, this is what my camera array looked like by park. So as you can see here, our highly urbanized park. I would like to note our 2 parks right here. This is Rock Creek Stream Valley, the one with 25 cameras. This is our smaller urban park, which is Sligo Creek. These are all highly urbanized. These two are highly linear. This one is more clumped in nature. And here towards the northernmost region, we have these big continuous areas parks of Little Bennett Regional and Woodstock Equestrian, while we have our suburban parks pretty well dispersed throughout. And so once we collected images from these cameras, birds were then sexed and ID'd. So toms, which refers to male birds, have the presence of a beard, which is a modified feather that protrudes from its chest. They also have brightly colored heads. Fun fact, the wild turkey was often regarded as America's bird because their head can experience shades of red, white, and blue. And lastly, they have brightly colored body feathers. Hens, on the other hand, are much smaller in size. They have dull colored body feathers, and they have the presence of a mohawk, which is a trail of feathers that runs from the base of the neck to the top of the head. So an occupancy and density analysis was done for all hens, toms, poults, as well as unknown birds. So a bird was classified as unknown if there was a partial image of the turkey, if the turkey was too far away to actually get an accurate idea of what its sex was, or if lighting was poor. And poults, which is everyone's favorite, are juvenile birds, and they were classified by being significantly smaller than females. So this imagery data was then put into a system called time-lapse, where we recorded here information about the park, the camera, temperature, and other potential landscape variables. And we also put things in like presence of turkey, the number of tens, toms, poults, as well as unknown birds. And for my analysis, there was approximately 463 data sets that we had to do all this for. So this was no small feat because most of these data sets were well over 1,000 images. So for my study period, I decided to choose two ecologically relevant periods for turkeys, one being the hunting season, which, as I mentioned earlier, runs from April through May. And so I divided the hunting season into 9-day subperiods of hunting early, hunting middle, and hunting late. Similarly, for the brooding season, which runs from July through August, I divided this into 9-day subperiods as well, for brooding early, brooding middle, and brooding late for both 2022 and 2023. I chose the brooding season because, as I mentioned, it is biologically relevant and we are able to distinguish adult female birds from juvenile poults. So an occupancy and density analysis was done for all birds across all study periods. And here are the total number of observations we had of turkeys across our camera sites. As you can see here, there's a high number of observations in this great Seneca suburban park. But I would like to draw your attention to Rock Creek Stream Valley, where in 2022, we had turkey observations at 4 different camera sites. Compare this with 2023 data, we still are observing quite a bit of turkeys at this great Seneca suburban park. But then if we look back at at Rock Creek Stream Valley, we have doubled the number of sites we are observing turkeys at. So we went from 4 turkey sites in 2022 to now 8 turkey sites in 2023. Remember, this is our highly urbanized park. So a little breakdown of occupancy modeling for my folks who may be a little stats phobic or don't know anything about occupancy modeling. It is actually very simple because it's all binary data. So you go out and you survey an area. You either see the species, which is assigned a value of 0, or you do see the species, which is assigned a value of 1. There can, however, though, be some contingencies when you don't see the species. So you either don't see the species and the species is actually truly not there, or you do not see the species, but the species is actually there and you just failed to observe it. For N mixture, which I am utilizing for my density models, here's the equation for it. I don't really want to get bogged down in the details. All you really need to understand is that it is expressed as the mean number of individuals at a site, and in our instance, the mean number of birds at a site. So for my occupancy and density analysis, I chose to overlay a grid. My detection matrices for both my occupancy and density analysis will be made at the grid level, and the number of cameras in each grid cell will be a detection covariate for my analysis. However, utilizing a grid sort of begs the spatial dilemma of to grid or not to grid. So one of the main model assumptions for occupancy and density modeling is that sites are independent of each other, and that I am not doubling counting a single bird across camera locations within the same time period. So by utilizing the grid method, I meet the assumption that by observing a turkey at site A, I am not then re-observing it at site B within the given time period. And so here's a close-up of my grid overlaid with Cabin John Regional Park. This is one of our urban parks. And my grid size was based off of what an approximate turkey home range was. So my grid cells are approximately 2 kilometers in diameter. And so I would like to draw your attention back to the occupancy modeling equation, particularly this middle column right here. So whether or not you do see the species or you don't see the species is essentially this weighted coin flip, right? Getting a heads or a tails, getting a 1 or a 0. And so what is driving this weighted coin flip? Well, in wild turkey ecology, it's things like land cover. Here I have Montgomery County overlaid with the Chesapeake Bay National Land Cover data set, and this data set is a meter by 1 meter resolution. Here again, we have Cabin John Regional Park with the camera locations in the grid. Similarly to how I'm building my detection matrices for my analysis at the grid level, I will also be extracting land cover data at the grid level as well. And so for my models, I decided to choose things that I thought were ecologically relevant to turkeys, specifically urban and suburban turkeys. So turkeys, as we mentioned, need these roosting areas. So I generated a covariate called Turk Forest, which combines the presence of forest and tree canopy other, as well as Turk food, right? We had mentioned that turkeys are these opportunistic feeders and they feed on a variety of things. So there was quite a few land cover covariates that got put into this Turk food model. I also included things like mean elevation, density of buildings, and density of roads in my analysis. These last two things sort of hint at some sort of human development or urbanization covariate. I also have additional covariates that I'm currently making in FragStats. This includes things like core area of forest, core area of cropland, and the interspersion of these two different land cover classes. Science is so humbling. So when I went to go do my occupancy and my density analysis, my models were not configurating. They were not converging. It was, for lack of a better term, a hot mess. And so when we look at things under the hood, it looks like we do not have enough detections of wild turkeys in our camera sites to be able to break up these analysis by sex and by age class with poults. So, which then inspired me to do a density analysis of the total number of birds. So I'm essentially combining the observations of hens, toms, unknowns, and poults to get this total Turk column, and that for the rest of my presentation, I went ahead and did my analysis on this column. So I currently have my occupancy code running. If you are really interested in my occupancy analysis, please follow me after this seminar to the spatial lab located just next door. But for my density results, here's how the models fit. So for 2022, in the brooding early period, Turk food as well as Turk forest were the best fitting models. For my brooding middle period, Turk food, Turk forest, and mean elevation were statistically significant. For hunting early and hunting late, it was both these Turk foods models that were statistically significant. Compared to 2023, brood early was Turk food as well as this density of buildings. Brood middle, hunt early, and hunt middle, Turk food was the only statistically significant model. So this sort of lines up with what we know about wild turkey ecology, right? Turkeys need food during the brooding season. In fact, this is their primary driver during the brooding season. As you can remember from the image earlier, hens are trying to feed their poults as well. So during the brooding period, it makes sense that turkeys are going to be in areas where there is a greater abundance or a greater presence of food. So in terms of management implication, this Turk food model best explains turkey densities throughout my 8 spatially unique parks. But what does this mean for urban bird management? So by understanding these results and having turkey food best explain wild turkey presence on a landscape, wildlife managers can then use this information to mitigate human-wildlife conflicts, one notable conflict being the spread of zoonotic diseases. Which then takes me to my second objective, which is evaluating site-specific turkey densities in relation to local tick abundances in 2 highly urbanized parks, which is Rock Creek Stream Valley, and Cabin John Regional. I hypothesize that locations with higher turkey densities will have greater tick abundances. So a quick little introduction on ticks. Females lay their eggs in the spring, and then ticks must complete 3 subsequent life stages: the larval stage, the nymph stage, and the adult stage. However, in order to move to the next life stage, a tick requires a blood meal, but what they get their blood meal from depends on their life stage. So, as you can see here, larval ticks often get their blood meals from small mammals like mice, as well as birds. Nymphs and adult ticks get their blood meals from larger mammals like humans, as you can imagine the implications for zoonotic disease spread there, as well as deer. The big question is, where do turkeys come in all of this? Well, we've documented nymph ticks on turkeys, and we've also documented adult ticks on turkeys as well. So for my analysis, I primarily focused on turkey data in these tick data in these 2 life stages. There are quite a few tick species native to the Mid-Atlantic region. However, when we think of ticks and turkeys, we want to think of 2 main ones: Ixodes scapularis, which is the black-legged tick, and Ambyloma americanum, which is the Lone Star tick. This one is known for causing Lyme disease. And so, as you can see here, I've provided the ranges of the black-legged tick and the Lone Star tick. The black-legged tick includes the entirety of the state of Maryland, as well as the entirety of the Mid-Atlantic region. Whereas the Lone Star tick here, you can see, includes all of the state of Maryland and most of the Mid-Atlantic region. Scientists hypothesize that as climate change continues to progress and areas continue to get more warm and more humid, that we can expect tick ranges to expand into areas that were once too cold or either inhospitable to these to ticks. So we can expect in recent years for this range to potentially expand. And so for 2023, the hunting and the brooding season, here is the number of ticks we've collected across our 8 parks. As you can see here, there is something clearly going on with Cabin John Regional, and I'll sort of circle back to this in a second. But one thing I like to highlight here is that this is not all of the tick data we've collected for 2023. This is just tick data that I've filtered to my 2 turkey ecologically relevant periods, which is the hunting season and the brooding season. So when we talk about the potential tick-turkey relationship, currently there are 2 conflicting hypotheses. For one, turkeys are thought to increase the presence of ticks in a landscape through their role as tick hosts. So a tick is feeding on a turkey, that tick then gets its blood meal and falls off into the landscape. Therefore, we can expect areas that have existing turkey populations to have ticks populations expand to as well. On the other hand, turkeys are thought to decrease tick abundance in a landscape. This is through their direct role through their dietary behaviors and their direct consumption of ticks. As I mentioned earlier, like we mentioned, turkeys are opportunistic feeders, so it is thought that they are actively consuming ticks from the landscape. So why should we care about ticks, and why should we care about this potential tick-turkey relationship? Well, Lyme disease is the highest reported zoonotic illness in North America, and throughout the 21st century, there has been increasing reports of tick-borne illnesses. And these urban and suburban areas are particularly vulnerable to zoonotic diseases because they exist within close proximity to wildlife communities. So as I mentioned earlier, there is an increased, Lyme disease is increasing throughout the 21st century. So the number of confirmed as well as probable cases is also increasing. And there's this huge health equity issue when we discuss ticks and tick-borne illnesses. I find this Springer and Johnson study to be fascinating because it discusses tick illness reporting in terms of things like social factors like race and ethnicity and socioeconomics. This study ultimately found that Lyme disease has a higher incidence of reporting in higher proportions of white communities and highly educated communities. However, we know that it is often marginalized communities that face the disproportionate effects of zoonotic illnesses, and they are also more at risk for severe health complications. So by understanding the latent mechanics of Lyme disease and other tick-borne illnesses, we can better promote health equity within our healthcare system. And I find this image really interesting. When we talk about Lyme disease, a lot of the physical manifestations of Lyme is documented on more Caucasian skin, but as you can see here on darker skin, the physical manifestation of Lyme is entirely different. So how did we quantify tick populations in Montgomery County? Well, we tick sampled 10 sites in Cabin John Regional and Rock Creek Stream Valley on a monthly basis. We had 3 transects at a camera that consist of a randomly assigned 30-degree ordinal angle. And this tick data, as I mentioned earlier, was filtered to the 2023 turkey brooding and hunting season. So tick sampling looked something like this. You get assigned your random ordinal angle. In this instance, it is 30, and our opposite angle is 210. You have a 1 meter by 1 meter denim drag behind you, and you are slowly walking through the woods, stopping approximately every 15 meters until you get a tick. So say you do get a tick. You then collect the tick off your drag and put it into a vial and give it an associated GPS point. And then you put your vial away and you continue walking your 100 meters until you reach this end point. Once you reach this end point, you then retreat back to the camera and then walk the opposite 100 meters. So we have 2 100-meter transects, which adds up to 200 total meters sampled, and we did this 3 times at each camera location. If we had to assign a winner for the tickiest park in this project, it would be Cabin John, by far.
Take a deep breath.
You good? Okay, cool.
I know, okay, all right, good to go. That way they can hear.
All right, you're good.
Do you want me to
It's
re-say
okay.
anything? Okay. So as I mentioned earlier, we've documented ticks on turkeys during the spring season, and we've documented ticks on turkeys during the summer and fall season. So really, is my filtering of this data to this ecologically relevant period to turkeys, is that ecologically relevant to ticks? Probably not, right? Since we've documented ticks on turkeys during this season, I can go back in terms of my analysis and sort of go, by unfiltering the data, look to see if we have more cameras where we have greater observations of turkeys and greater tick data, and then sort of move forward with my analysis there. So in terms of moving forward, I still need to merge my observations of turkeys from my 2022 and 2023 occupancy analysis, as well as adding the site-specific tick abundance covariate to my N mixture model, and keeping in mind that I am not going to be filtering this data. And so one thing that I would like to add that's not in terms of science at all is that I'm extremely grateful for the opportunity that I've had in order to work in Montgomery County and work with the wonderful people in the ASWEL lab. I am actually from Montgomery County, so I grew up right in Muddy Branch Park. As you can see, that's a picture of me and all my siblings as our childhood home. And I went to high school in Montgomery County. I ran in Muddy Branch. I ran cross-country races in Little Bennett. I lost my first tooth in Cabin John. So I've had this really awesome experience of being really intimately tied to this project, in the sense that this is where I grew up. And it's been such an amazing experience to grow up being this huge wildlife nerd, to now be able to analyze wildlife communities in my own backyard at the graduate level. But no project is a great project without the people on it. And so I have a few people I would like to thank. My UMD wildlife community, which is the ACEWELL and Urban Nature Labs. You guys are awesome. Thank you so much for the support and fun jokes you have provided me with this time. I would also like to thank my advisor, Dr. Jennifer Molinax, for her mentorship. I would also like to thank my mentor, code wizard, friend, Dr. Matthew Gonerman. He has been incredibly patient with me throughout this process, and this project ultimately would not be possible without him. I would also like to thank my committee members. I'm very lucky to have one sitting with us here today, Dr. Travis Gallo and Dr. Franny Buterman. She is at Penn State University. And raise your hand here if you have processed an image set for turkeys. Hannah, Trinity, Allison, thank you guys. Can you guys offer me a round of applause with these individuals? Like I had mentioned earlier, my analysis alone was 463 data sets. These data sets could be 6,000 pictures of wind with the occasional turkey walking by. This was a very, very long process to get this data cleaned, and I'm extremely thankful for all of their hard work and hours put into this project. And without further ado, any questions for me? Yes.
Venture in, not knowing much about wildlife.
Totally fine.
So, do you have a sense of sort of the significance of turkeys as hosts compared to other mammals that would be occupying these and their functioning as hosts? And I mean, I know you've got this turkey data set, but is it possible that there's something else that sort of dominates?
Yeah, so when we look at the tick-turkey relationship, there is primarily this one paper, which is this Osfeld et al. 1999. So in terms of the scientific literature, there's been very few studies examining this tick-turkey relationship. Are ticks documented more on turkeys than they are on deer? I would say probably unlikely, because we know deer are a huge host for ticks as well as small mammals. So as to whether or not turkeys are as important of a host to ticks like deer and mice are, I would say probably not. But like I said, there really hasn't been much scientific literature about that. Yes.
Do you have thoughts on why Cabin John has such a higher prevalence of ticks than the other parks that you looked at?
That's a great question. Cabin John is unique in terms of our highly urbanized park, is that it has this large core area compared to Rock Creek Stream Valley, which is this highly linear. I don't know if like the core area versus the highly linear has anything to do with it. It probably has something to do with there's more suitable tick habitat in these locations. Yes.
On the basis of occupancy framework, you said that you had a cell size of 2 kilometers.
Yeah.
Can you give me an idea about the homelands of turkey?
Yeah, so a wild turkey, there's this one paper that says a wild turkey home range runs anywhere from approximately 1.5 to 3 kilometers. And so I chose 2 as a safe boundary between those two extremes. Yes.
So the turkeys have the roosting and the brooding.
Mm-hmm.
So maybe they have the ticks, mostly you're sampling out the herbaceous.
Yeah.
There. It looked like there's a lot of variation in that. Like in one of the first pictures we had, it looked like Japanese stilt grass.
Mm-hmm. Yeah.
I don't know. So
This one?
I just took a quick look. I said ticks don't like Japanese stilt grass, but some plants I think maybe ticks prefer more. And you see variation that like that.
This,
picture, you see
yeah.
that sometimes in urban areas and floodplains, and I guess I'm just wondering how the understory might affect tick and brooding co-occupancy, if you will.
Well, in addition to the occupancy, analysis that I'm doing and the tick sampling, our lab did an entire in-depth vegetation survey at all of these camera sites as well to sort of document at our tickier sites, what are the vegetation specifics, right? Because we get this land cover data set, this 1 meter by 1 meter, but that's not telling us specifically if there's invasive vegetation at those sites. So we did that as well. This image you're referring to, which is Great Seneca D, is actually very interesting because we have a high number of observations of turkeys this year, as well as that this site is also extremely ticky. So who knows? Does that answer your question?
Is that stilt grass?
Yeah, it's Japanese.
Yes, that is Japanese stiltgrass.
So they do like stilt.
They do.
They do. Yeah, and primarily when you go out and you look at this site, this is a very open site, and I'd say for like a good 100 meters by 100 meters, it is primarily Japanese stiltgrass.
Ticks really love this.
They do. They really like this site. Yes, Dr. Lisa.
So Maddie, thanks for a really interesting talk. I have a kind of analytical stats question.
Oh, geez.
So it looks as though you analysed each of your seasons separately.
Correct.
Would a more elegant approach be to combine all your data sets and look at interactions between seasonality and landscape variables? Or would that just create a model that would be too unwieldy? Or what was your decision regarding that?
So my decision was primarily based on what we've seen in previous turkey literature, right? And I primarily did separating them into the brooding and the hunting season as means of comparison, right? When we look at turkey occupancy and density studies, it primarily is comparing them against these two ecologically relevant periods. So that was sort of my decision in terms of moving forward, is that it's just been what's been previously done in turkey studies.
And that results in a number of different tests, right?
Yes, correct.
Do you apply a correction or so? P-value, just so that you don't get false positives? It could be no.
Correction value. Yeah, I haven't, well, I'm trying to think of everything that I've done with my code, and I can't say I've done a correction value. I can, though, add specifically with the covariates when I built that turkey forest and that turkey food model, that I ran a correlation test on the different land cover covariates to see if any of them were highly correlated, and then that wasn't, those land cover variables that were highly correlated were not included in the same model. So, but no correction factors that I know of.
Thanks. That was just the tricky stats.
I appreciate it. It made me think. Anything else? Yes, Colleen.
So you said that there are lots of different tick species that exist in this part of the country, in your study area. Why wasn't it that you actually just chose those two species?
Because that, based on what the very small literature we've seen on ticks and turkeys, it primarily focuses on black-legged ticks and these Lone Star ticks. There is no real justification, based on what we've seen ecologically in previous literature, to add things like dog ticks into there. When we think of turkeys, previous literature has really focused on the Lone Star and the black-legged tick. Yes, Carson.
Thank you, this is fantastic. I
Thanks.
think we as graduate students are always feeling the pressure of time and feeling rushed and things, so if you had more time, what's something that maybe you would have done differently or something that you wish that you could have done?
Can I add if I had more time and more money?
Sure.
If I had the money, what I think would be really interesting is putting backpacks on urban turkeys to get more fine spatial data in terms of how they are specifically using these urbanized areas, and to then sort of estimate their home range in these urban areas as well. I think that would be fascinating. In terms of my project, I feel very complete and at peace with it. I really enjoyed this project. This was a really awesome experience for me. Yeah. Yes, Dr. Lisa.
Okay, Maddie, given that you've worked with turkeys, do you think that we should, as a country, replace the bald eagle?
100%, absolutely.
Because we don't really do any research on bald eagles in this.
We don't, and yeah,
I would eat a deep fried bald eagle.
yeah.
It's like
Isn't there like Modern Family or something where it's like Thanksgiving and the kid comes home with a bald eagle? Is there something that I'm remembering? Yeah.
This is the, like, birds eat fish are very tasty.
Turkeys are very tasty.
People think that way about carps, but you can carps.
Is there any chat questions?
Oh yeah, is there any questions in the chat?
No, there aren't.
Those are all from us.
That's awesome. Thank you to all the folks who attended on Zoom. Any last final thoughts? Yes.
Can turkeys get Lyme disease?
I don't think that's been documented, no. So Lyme disease is often commonly misrepresented. So ticks actually get Lyme disease from small mammals. They don't get it from deer. So in order to contract Lyme disease, you have to have specifically a tick on you that has once fed on a small mammal and has contracted the Borrelia burgdorferi bacteria.
So Peter May had a comment that's kind of interesting. He says turkeys lost by 1 vote to be the national bird.
1 vote. Colleen, did you have something?
Yeah, I was just curious, because I think other people in your lab are working on small mammals and ticks.
I wonder who.
What kinds of, like, how does your research support what they're exploring, or might?
Yeah, so Carson's, Grace's, and I research is sort of all this big collaboration, right? So Grace is looking at wildlife communities in Montgomery County. I'm more specifically looking at the turkey community in Montgomery County. But Carson is looking at small mammals and diseases in these 2 highly urbanized parks. But all of our projects really overlap with this tick data. And it was this massive effort by everyone in the lab to go out and collect this tick data. And it was by no means easy feat. To collect ticks, you have to go out at the hottest part of the day. It couldn't have rained the day before. So there is sort of like this time aspect thing that goes into it as well. But it really is just primarily this tick data. I don't look in terms of like zoonotic disease spread from ticks to turkeys or turkeys to ticks, so. Anything else?
All right, well, thank you, Maddie.
Thank you.